Hello student, because the lecture this week is cancelled, I decided to record this video. Hopefully this will be helpful. This video is going to include the following components. We're going to briefly talk about the graduate project, the exam 1, and we're going to review what we learned last week, which is a primer design. And we're also going to briefly talk about cell culture technique and cell signaling. And these two are going to be helpful for your Campari cell culture project. This is our roadmap, and if you look at this map, and you will find that you already had accomplished a bunch. And last 2 weeks, and you have amplified this Campari gene through PCR, and you have also done the restriction digestion of the PN34 vector. The lab that is cancelled this week, we are going to run a gel to visualize this PCR product as well as PN34 vector digestion. Because the lab this week is cancelled, so we are going to incorporate part of this lab into the lab 5. For 510 graduate students, and you probably should be able to notice that on the Moodle site there are 2 folders, and one is bit 510 grad project documents, one is a graduate cloning project. This grad cloning project contains a graduate project lesson that can help you to design your cloning project. In this lesson, we will walk you through the steps of cloning your gene, and you can select the gene that is interested for your research or the gene that you want to do research in the future, and you can select a vector. You can design a cloning strategy that how you can clone that gene into the vector. So based on what we have learned in the last few lectures, and you should be able to start working on the graduate project now. Exam 1, and I already sent out several emails earlier about exam 1. So basically the exam 1 have 2 components. One is a take-home exam, and one is individual exam that you would do at the Delta Testing Center. So the take-home exam 1, and you can have access of the take-home exam 1 if you go to the Moodle site, and you can go to week 5. On the week 5, there is a session here called exam, and if you click into the take-home exam 1 individual submission, you will see 2 documents. One is a Word document that you need working on. The other is this vector map that you will need for working on this exam. After you finish the exam, and you should be able to submit in this week 5 folder. After you submit, after you give your individual submission, and then you can working on a group that we have assigned, and then you can submit your group submission on week 6. So today we are going to talk about cell culture technique and hypothesis design. This picture here is showing a cross-section of the EM image of yeast cells. You can see really cool. You can see the cell nuclei and the intracellular components and cell membrane. So before we dive into the cell culture, let's briefly review what we have learned last week, which is primer design. So let's review that by looking at a question. So assuming that you have a sense sequence that you write from 5' to 3' here. In this region that you have highlighted here, this is the region that you want to design your reverse primer. And the reverse primer that you design, you want to incorporate the GAA TTC restriction site. And how are you able to do that? So before I reveal the answer, and please take 1 minute to think about the question and write down the answer that you think is correct. Okay, now we have our sense sequence. We want to design our reverse primer. First, where the reverse primer is going to bind? Yes, the reverse primer is going to bind at our sense sequence, and the sequence of reverse primer are going to be complementary to this sense sequence. So we can write down that, which is CC, GT, CGAA, CGTT, ACC, AATG. So for the sense sequence, the left is 5' and the right is 3'. So for the complementary reverse primer, it will be opposite. So this will be the 5', this will be the 3'. Next, we want to incorporate this GAA TTC restriction site to this reverse primer. If you still can remember what we learned last week, the restriction site will be incorporated to the primer as a 5'. So let's write down this GAA TTC restriction site at a 5'. So next, the reverse primer. So the primer you should write at the beginning. If we write the primer, we write the beginning at a 5' and end at 3' from left to right. So we need to flip this primer and let it 5' to be on the left. So the primer we designed will be this sequence in the end. So finally, and as we learned last week, to make a restriction enzyme work properly, you need to get a hold on some short sequence. So we cannot let the restriction enzyme working on the edge like this. So to make the restriction enzyme hold some short sequence, let's add several A's to allow the restriction enzyme to work properly. So after all the explanation, and you will know the right answer for this question will be C. So hopefully you got your answer all correct. So the learning outcome for the lecture this week is going to include, we're going to use some web resources to identify some useful information. We're going to compare the contrast prokaryotes and mammalian cell culture, and we're going to focus on the cell signaling, especially the calcium signaling. We're going to walk through some examples to look at how to design a testable hypothesis and design experiment to test that hypothesis. This image is showing the prokaryotes and eukaryotes, and in our projects we are going to express and the Campari gene in both systems, and after we introduce the gene into the E. coli cells and CHO cells, and we want to culture both cells and to allow this gene to be expressed. The cell culture technique in the prokaryotes and eukaryotes are a lot of similarities and also a lot of difference. And here we're going to look at the compare of these two systems and how. So first, in the prokaryote system, and in our case, we're going to use E. coli, and mammalian cell culture system, we're going to use Chinese hamster ovary cells. So for the E. coli cell culture, we can either culture it in the liquid environment or culture it on this LB plate. And the mammalian cells, and we culture it in the liquid cell medium. Both prokaryotes and mammalian cells need nutrients to grow. In addition, we need dissolved salts. And for mammalian cells, we need dissolved salts that mimic interstitial fluid. In addition, we also need other components that are found in blood. For the E. coli culture, and we need to supply them with oxygen, and usually we just culture it using the air, oxygen. And then for the mammalian cells, they need to have a appropriate level of oxygen and CO2. So this needs to be controlled. The pH for the prokaryote cell culture is usually around 7.5, and the culture of prokaryotes and E. coli need to be at 37 degrees. And this is the same for the mammalian cell culture. So to be able to culture both of the systems can successfully, and you want to only introduce the cells that you want to culture, and you do not want to contaminate the cells that you culture with other cells, for example, some bacteria in the air. So for that to be able to work properly, you need to have the specific sterile technique. For the prokaryote cell culture system, usually we'll use a Bunsen burner, and around this fire region, and this will be sterile, and so you can use a capped tube and to culture your bacteria here. And for the mammalian cell culture, we'll perform it into the biosafety hood. So since we have already introduced this, we are not going to explain this further. This image is showing that in the in vitro system and the in vitro system and the in vivo system, how the neuron cell will look like. Besides you will discover how pretty those cells are, and you will also find how strong connection each neuron cells with the nearby cells as well as its environment. Similarly to humans, we need to communicate with each other. We need to respond to our environment, and cells also need to respond to their environment as well as communicate with each other. So for right now, and you are listening to my lecture, and you use your ear to perceive the signal, and you think about it, and after processing it, you either think what I said is good, or you think that is trash, and you make a response, either write it down as a note or discard it. So similarly, the cells on the membrane also have a lot of ears. They are able to hear and the environment, and those ears are called receptors. So the cells can detect and respond to their environment through the mechanism and through the cell signal transduction mechanism, and usually we can divide it into 3 steps. So step 1, the cell can perceive a signaling through the reception, and step 2, the signal that perceived by the receptor need to be transduced and within the cell, and finally the cell will make a response and to the signal that it received. So the step 1, reception. The cells have proteins called receptor that can bind and to the signal molecular. Usually those signal molecular are called ligand, and there is a specificity for the receptor-ligand interaction. So usually when a ligand binds with a receptor, it will activate this receptor. One of the receptor example is a G-protein coupled receptor. So G-protein coupled receptor is the 7 transmembrane receptor family, and it can bind after bind with a signal molecular, and it can activate this small G protein is associate. So after bind with signal molecular, and this receptor will have a open domain that can interact with this small G protein. The interaction of small G protein with this GPCR will induce the change of this GTP to a GTP that will activate this small G protein, which then can further activate the downstream signal. This is how the receptor will look like, and you probably will think that GPCR, what is that going to do with me? Actually, GPCR have a lot to do with you. So a lot of the receptors on your olfactory neurons, or a lot of receptors on your taste bud cells, or a lot of receptors on your intestine sensory cells, they're all GPCRs. For example, the fact that you can smell and the fact that you can taste the fatty food, can taste a lot of sweetness, and they're all due to the GPCR. So next time when you try to eat a cake, and you can stop and think about, I thank for my GPCR to allow me to enjoy the delicious cake. After the GPCR perceive the signal, it needs to transduce that signal downstream. So the GPCR usually uses second messengers. Second messenger are small molecules that initiate and coordinate intracellular signaling pathway. In this case, this signal molecule, and which is a ligand, combined with the GPCR, and then can activate the downstream small g protein called small g protein. And then activated G protein can then activate the downstream phospholipase C. The phospholipase C then can act on the cell membrane lipid molecule and induce the release of this small molecule called IP3. So the small molecule IP3 can then act on the ER membrane-associated calcium channel and then induce the calcium release from ER to the cytoplasmic, and then the increase of the intracellular calcium can activate associated downstream signals. So besides IP3, there are a lot of other second messengers can be associated with GPCR. For example, the CAMP, DAG. Finally, after the GPCR and perceive this signal and transduce the signal through some other downstream signal molecules, the cell needs to make a response. The response may vary, and response can either change in gene expression or activate or inhibit another signal pathway, or the response can be secrete molecular and the cell can move and can grow, divide, or even die. The second example of receptor is ion channels. One example of ion channels is the calcium-associated ion channel, and this is one of the classic examples is the calcium ion channels that will be activated during exercise. The activation of the calcium channels and on the skeletal muscle cells is very important and for the skeletal muscle contraction. The third example of receptor is this receptor tyrosine kinase family. So kinase, if you look at it, this is some of the enzyme that can introduce a phosphate group to the downstream substrate. One classic example of a receptor tyrosine kinase is this insulin receptor. So the insulin receptor expresses in a lot of tissues in our body. One of the tissues the insulin receptor expresses is our adipocyte tissue. So after we eat and a lot of sweet things, and the sweet and the glucose in the blood can stimulate insulin release from our pancreas. And the released insulin can then bind with the insulin receptor on the adipocyte. And activate of this insulin receptor can then activate multiple downstream pathways. One of the downstream pathways will tell the adipocyte to translocate the glucose transporters to the cell membrane, then can allow the adipocyte to eat a lot of glucose. And those glucose into the adipocyte will then be transformed into the fat. So next time when you stand on a scale and you notice that the number is increasing, and you can blame the insulin receptors on your adipocyte. So in summary, and how can cell detect the response to the environment? The cell can through the steps of a reception, and need to have a receptor to bind with signal molecule, and cell can transduce that signal to downstream to another a lot of downstream signal molecules, and cell can respond and to the stimuli. So how can chemicals like a drug and will interact with this pathway? So the chemicals can either activate this pathway or inhibit the pathway. So the drug can either become agonist, which means that the drug can bind with the receptor and activates the receptor downstream pathways. Or the drug can be antagonist, which means that the drug can still bind with the receptor, but binding with the receptor do not activate downstream pathway, and because the drug binds with the receptor, it blocked its native signal molecular bind with the receptor, so the whole downstream signal pathway will be blocked. So after all those introductions, hopefully you will feel better about something like this. Actually, in our body, the cell signaling pathway is not linear. It forms a very complex network. So each of the cell signaling pathway will influence other cell signaling pathways. So today we are going to talk about calcium signaling, and calcium signaling is one of the important and intracellular transduction small molecular signaling pathway. So the calcium signal, they have a gradient. Outside the cells, the calcium is usually really high, usually at 1 to 10 millimole concentration range. But inside the cells, it's really low. It's about 100 nanomole. This is about 100,000-fold difference between the inside cells and outside the cells, and why the cell need to do that. And besides that, you will find that in a lot of intracellular organelle, like this ER and mitochondria, the calcium concentration is also relatively higher. And which means, so altogether, and we will say that calcium is an important transduction signal molecule, and calcium and have a gradient and is compartmentalized. So in our first lecture, we had already introduced the calcium signaling can mediate a lot of responses, and this response can include the exocytosis and contraction of muscles, and can influence metabolism, transcription of gene, can influence the cell growth. So let's look at one specific example of a calcium signaling modulation by GPCRs. So before we look at that specific example, and again, let's reintroduce the idea that the calcium signal have the calcium concentration have the gradient. So outside the cells, the calcium concentration is really high, and the intracellular cytoplasmic cell's calcium concentration is really low. And when the cells need to be activated, and the intracellular cell calcium concentration get much higher. So the cells try to keep its resting intracellular calcium concentration low, so that when there is a signal molecular stimulation, the intracellular cell calcium can really increase a lot. So the cells keep the intracellular calcium concentration low, and through a lot of cell membrane associated pumps, and those pumps are very busy to pump the calcium out from the cells, and the pumps can either located into the cytoplasma or located in the ER. So in the ER, the pumps can pump the calcium from the intracellular components and to the ER. And the calcium pump and at the cytoplasmic can pump the intracellular calcium from the cell and outside of to the extracellular components. So within signal molecular stimulation, and a lot of receptors can be perceived as stimulation. For example, and the signal molecule combined with the calcium channels, and which can activate this calcium channel and induce the calcium influx from outside the cell into the cells. All the cell signal molecule combined with the receptor, like a GPCR or receptor tyrosine kinase, and induce a secondary molecular release, like IP3. The release of IP3 can then bind with IP3 receptor on the ER, and then induce the ER calcium outflux from the ER to the cytoplasma. So many pathways can mediate intracellular calcium, and let's give an example of AP3 mediated calcium signaling. First, we will have a ligand that combined with the GPCR receptor can lead to activation of second messenger like AP3. Then the AP3 can activate the AP3 receptor located on the ER, and then which can induce the AP3 gated calcium release. So the calcium flux out of the ER and into the cytosol. The calcium combined to the calmodulin domain of an effector protein, causing it to change its shape. And the shape change of the effector protein ultimately causes a cellular response. Finally, and after the calcium activation, this increase of the calcium need to return back to normal. So that require those protein pumps that either work on the cytoplasma membrane or the ER membrane and to pump those calcium back. So after all that introduction, hopefully you will have some more ideas about how to modulate a calcium signaling. So today, besides the cell signaling, we are also going to use a specific example to talk about NCBI database and related web resources. So NCBI is short for National Center for Biotechnology Information. This is a government-supported or maintained resources for biomedical or genomic information. It's really a powerful search engine, contain a lot of resources. And the example we are going to use today is some chemicals that contained in the red chili pepper called capsaicin. This is the chemical structure of the capsaicin molecule, and we want to identify, and as we all know that eating the chili pepper or spicy food can induce a lot of uncomfortableness, and that we can either call spicy feeling or hot feeling. So, but do you want to understand how the hot feeling getting produced? So to do that, and let's use NCBI website. First, we can go to the NCBI homepage website. On the NCBI homepage website, on the right, you will see a lot of popular resources. The PubMed is a really good resources for the literature searching, and the bookshelf, you can find a lot of free books about molecular biology, cloning, or biomedical resources. And the BLAST, we are going to briefly talk about today, and this is a kind of a software tool allow you to align your gene with the database. NCBI nucleotide genome or single nucleotide polymorphism and a protein, and another popular resources PubChem. So first, we want to see that how capsaicin work, and we can click the PubChem, and we can input our single molecular name capsaicin, and then click go, and this will come with the information of these associated chemicals. And on the PubChem web page, you will find the information about this molecule, the structure, and drug information, pharmacology. And you will find a lot of basic chemical information about this molecule and some of these brief summaries. So for us, in this search, we want to identify the target of the capsaicin component. So to do that, let's go to the pharmacology panel, and in this panel we can find that capsaicin is a TRPV1 receptor agonist. Okay, so we find capsaicin actually acts on a protein called TRPV1. So TRPV1 is a transmembrane receptor ion channel complex. And then we can also find that this TRPV1 not only can be activated by capsaicin, but also can be activated by high temperature. So that kind of makes sense. That's why that the feeling that you eat this spicy pepper can also be interpreted as a feeling of hot. So now we have identified the target TRPV1, and how can we further understand this TRPV1 receptor then? So to do that, let's go to the NCBI gene. You can just Google NCBI gene and it will lead you to this web page, and then you search TRPV1. Search. And then it comes with the TRPV1 genes from different kind of organism, like from the mouse, rat, human. Let's first look at TRPV1 in human. So on this information, we will through the NCBI gene web browser search, and it will lead you specifically to the TRPV1 gene information. And this on the right section here, we showed you the summary of the whole web page. You can find a summary of this gene, you can find the genomic context and the expression pattern of this gene. This is where the gene was located in the chromosome, the expression pattern, and other published articles in the PubMed about TRPV1. And after that, we can also find the general protein information and NCBI reference sequence. That's a sequence that we can use to identify these are the gene sequence information, which is this sequence number, or the mRNA sequence information, which is start with NM. So this arrow here was showing that this TRPV1 mRNA start with this NM number. We're going to translate into this protein and the protein sequence, and also have associated number that start with NP. So let's first look at this mRNA that from this TRPV1 gene. So we can just click this NM associate number, will bring you to another web page that showing the human TRPV1 mRNA. So you can see all the information about this mRNA, and you can see the translated protein from this mRNA, and this is the mRNA sequence. So after we find this mRNA information, and the next thing we can do is run a blast and look at whether specifically in the Chinese hamster cells, which is mammalian cell culture model that we are going to investigate, and whether the Chinese hamster cells, do they express this TRPV1 gene or not. So you can do that by click the run blast, and this associate mRNA NCBI reference number will automatically be added here as the enter query sequence. And here you can find a section called organism. So you can input your search is a Chinese hamster. So you can select Chinese hamster as an organism, and you can run the blast. So this blast, what it will do is it will use this as a query and as a search, and to identify within all the Chinese hamster genomic sequence, and whether they can identify similar sequence as this human TRPV1. So let's submit. Let's wait for a while when they proceed. So what the information can tell us? So the information can tell us whether, okay, the Chinese hamster expressed TRPV1. In another setting, it can tell us whether the Chinese hamster can taste the spicy food or not. So okay, here's some result come out, and this is our submitted human sequence here, and this is the Chinese hamster sequence. You can click it and you can see some information. You can see the information down here. You can see there is a Chinese hamster, and TRPV1 cannot predict the sequence. And with this sequence, this is the associate number. So this is the academic name for the Chinese hamster. So you can find the associate number for the TRPV1 mRNA in the Chinese hamster start with XM. So you can click. So this is the highest kind of identified, most similar sequence of within the Chinese hamster genomic sequence, and within that Chinese hamster database, this is the most similar sequence they can identify. Click. Okay. So now we did find a TRPV1 in the Chinese hamster. So the next question is, what is the function of TRPV1? So I also put the whole search process here. The next question is, what is the function of TRPV1? To do that, and you can either go through, you can either look at what you have found in the NCBI gene, the TRPV1 gene web browser, and or you can go to the literature search through either Google Scholar or PubMed. So both are very powerful search engine for especially the biomedical field research. So when you do the literature search, you will find two types of articles. One is the review articles, and one is the primary research articles. Here we're going to give an example of the review articles and of the TRPV1. So for example, in this article come out is summarize the TRPV1 receptor in the 10 years, and from China cloning and to antagonist proof of concept research. This is a review article focused on the drug design. So you can read through it, and the review article, which is nice, can give you a quick summary of what is happening in this field, quick summary of this gene, quick summary of what other people have done, but also but one thing we need to pay attention to the review articles is because it's a review article, so it's summarized and by certain number of scientists, so it do have the objective view. So some of the view need to be taken critically and within the review article. So another one is the primary research articles. Again, this is some example of a primary research articles about TRPV1. So in this article, it come out as a activational calcium signaling through TRPV1 by nNOS and pyroxene nitrate as a key trigger of skeletal muscle hypertrophy. So you can see the title is really specific. So talk about TRPV1 can be activation by some specific chemicals and in a specific model, which is skeletal muscle. So the primary research articles usually contain abstract, which is a brief summary of what they found, and have an introduction of why they want to do the research, as well as the results, and usually contain multiple figures and can demonstrate the results. And after the results, and usually will have a discussion but to discuss summary and discussion, to discuss what they have found and in this study, and the result from this study, how that connected to the bigger field. So finally, in the primary research articles, you will also find the methods and the methods region, which will usually have a bullets of all the methods that have used in this study, and this is to make sure that other researchers, when they refer to this research, and they will be able to reproduce. Okay, so with that in mind, and apparently you cannot break and find your research partners right now if you listen to my video today, and hopefully you still will be able to get a copy of the worksheet and print out, and working on that. So before we jump into the worksheet, and let's briefly talk about the scientific method. So usually when you want to investigate in a research usually starts with a question, usually a very good question that usually make you excited and want to answer. So first thing you want to do about research is not immediately jump into the experiment. You want to do a lot of background research, and you want from the question come with a good hypothesis, a testable hypothesis. From the testable hypothesis, then you design your experiment to test that hypothesis. Through the result of the experiment, you either can get a positive result, which can prove your hypothesis, or you get a negative result, which is against with your hypothesis. So a lot of times, and this circle needs to be continual, and that's why the research is called a research. So a lot of times after you conduct a successful series of experiments, and you can write your research as a paper format or present in a conference, then you can communicate with your research results with other scientific members. So in this little Campari research project, and today, and what you can do is you can start by asking a question and come with do your background research and construct a good testable hypothesis for this Campari research project. So after you come with a good hypothesis for this Campari research project, the next thing you want to do is to design a good experiment. So when you design a good experiment, there's several terminologies we want to introduce. First, research questions, and hypothesis, and experiment. This is some procedure that you want to conduct to test your hypothesis. An assay is something you need to run or test or run in an experiment. Independent variable, and usually will be changed in a controlled manner. So in this case, we want to test a specific drug and how that drug going to influence the calcium signaling. So that drug is going to be your independent variable. Dependent variable will be some value that you're going to be tested and measured. So in this case, that the effect of that drug on the calcium signaling. So the calcium signaling is some result that you are going to be measured. The confounding variable is something other than independent variable that could affect the dependent variable. So for example, in our case, when we conduct our Campari research, we give the cell the drug that you selected, but however, they still have a lot of other variables, for example, the temperature, and or even which day that you conduct the research, or the condition of the cells. All those can influence your result, but that is not what you designed to be changed. So we can also conclude our Campari research project using this diagram, and in this semester we are going to introduce the Campari gene to the CHO cells, and then we're going to express the Campari gene in the CHO cells. What the Campari do is an indicator of the intracellular calcium concentration. So we can use this cool system to measure the intracellular concentration. Then we can add a drug that we selected, and through conducting the background research, we can select a drug that we're really interested. We can use this Campari system to measure the intracellular calcium level. So by doing that, we're going to measure if you can still recall from the first lecture, how the Campari be able to indicate the intracellular calcium level is when at a high calcium concentration state, and the Campari gene is going to convert from green to red under UV light condition. So again, you can think about all the terminology involved in this research experiment design. So one thing that we want to pay attention to is within the experiment design is in order to make sure that our results can lead to the conclusion that can lead to a defined conclusion, we will usually also need to include a positive and a negative experiment control. For example, in one experiment, you want to test one specific fertilizer, whether it can promote the growth of a tree. What you can do is you can use a positive control, you can use a known fertilizer that is known to work and to fertilize that tree, and you can treat another tree with your experiment and a fertilizer. You can treat a negative control group, which you only water the tree without any fertilizer. Then you compare all the three groups. So in the ideal situation, this positive control, you will see an increase of growth and upon this known fertilizer. And if your fertilizer is actually promote growth, you will also see an increase of growth when they compare with the negative control. So in our experiment, similarly, we want to introduce a positive control, which is a known calcium activator, and which is ATP molecular, and we are going to introduce the cells with our experiment group, which is the drug that you are going to select. We are going to also have the negative control group, which is only the vehicles like DMSO that okay. So in this research projects, and we are going to provide the panel that on the right, all the FDA approved anti-cancer drugs, and we are going to test how all those chemicals are going to influence intracellular calcium by this Campari CHO cell model. So what you can do is you can follow the worksheet and do the background research. As an example that I introduced with the capsaicin, use the NCBI or PubChem or PubMed resources, and then you can look at some of the drugs that you selected, look at their information, and choose the top 4 drugs that you like most. And then you can do a background search on those drug targets. Once you identify a drug, you should be able to see the drug targets, and then you can see how that drug target is going to connect with the calcium signaling. So then you can predict whether treatment of this particular drug on this CHO cell model is going to increase the intracellular calcium or decrease intracellular calcium. So sometimes you will find the drug can act on some drug target, like in this case the VEGF receptor that is directly linked to the calcium signaling here. So this is more straightforward. However, sometimes you will find some drug that act on some drug target, like in this case cyclone D or CDK, which is important cell cycle modulator that do not have a direct link, appear to be to the calcium signaling. So in this case, what you can do is you can look at the downstream signal and how that cyclone D or CDK going to influence. And instead of look at direct link of cyclone D with the calcium signaling, you can look at whether this signal molecule, the MEK or SMAD3, is going to have any interaction with the cell with a calcium signaling pathway. So after you're working with your group this week, and you should be able to come with a top 4 drug that you select, and you need to submit that top 4 drug selection by next week, by the lecture of next week. Actually, I think you should be able to submit the drug and top 4 drug selection by next week. And then following that, and you're going to working on a progress report, and you have 2 progress reports. And then for this CAMPARE project, you also need working on the summary, which is after you have conducted your CAMPARE experiment. And then finally, you have a reflection and how this CAMPARE project had helped you to understand all the concepts. So this is the kind of a due date and the component of each report or the executive summary. So the worksheet that we provide is really going to help you to start this process by looking at the background information and help you come with a testable hypothesis and look at the primary research articles and search for the relevant materials and methods. So one thing that you may want to use during your writing, when you write this report or the summary, is the citation manager software. So the NCBI provides some citation software like RefWorks, and you can really use it to help you to insert your citation. So in this week, and I hope you will have time to working on the worksheet with your group members, and each group you will submit 4 of the components, and so each group and usually will have a 2 lab station, and in this 2 lab station you will perform an experiment, and which means that this 2 lab station, you will be working on the same drug, but one condition about the drug can be variable. For example, you can choose working on different drug concentration, or you can choose on different incubation time. So we are not going through the worksheet right now, and I already give you an email in our previous email. I already give you the item of the deadlines for the upcoming items. So hopefully you can keep those deadlines and keep on track. So that's all I have today. So try to be safe, and I hope you have a good weekend, and thank you. Bye bye.